So welcome back again. So we are on the class two of genetics PYQ discussion. Okay. So there is one question usually which is asked and it is most likely to be asked in the 2023 also. PSC mains. Why is it relevant? Because you know about the GM mustard being now being cleared by GSC, and it is said that in uh, three crop seasons this can come into cultivation with the approval of Ministry of Environment. Now, just only one thing is remaining. If Ministry of Environment gives clearance, then we will have GM mustard. It is a reality. Okay, till now only we have cotton. So, what is this transgenic mean stability? Listen to it very carefully and understand. Sure. In case of transgenic mean stability, we are using genetic engineering technique. Okay, we are using genetic engineering technique to transfer genes which are useful for bringing about male sterility. So you will ask me, so is it there in all the crops? No, no. We are using this transgenic male sterility in crops wherein there are some issues wherein there is no good male sterility system. For example, in Bajra, we have very established CGMS system like 5023A we have, and many cytoplasm, A1 cytoplasm, many A2, many cytoplasms. So there we do not require the much of what do you say this transgenic approach. But in the mustard and all, we have no good established this one CGMS systems. So we are going for transgenic. So this is similar to this GMS. This is CGMS and this repetitive. Okay. And what we have done is we have used the species Bacillus amyloliquefaciens, okay, as a source for these genes because these are transgenic genes. These sources we use. So what we have done is this Barnes genes is integrated into A line. A line means this will be made into male sterile line. Okay, for this male sterile line, we are integrating this Barnes. Okay, okay, and for this B line, or we call better we call it like the restore line, that is the R line. Okay, in case of this R line, in the restorer line, okay. So what we are doing, the R line. So here we are introducing the bar star gene to this system. Okay. So what happens in this bar star? Okay, this is maybe better to define this way. Okay. So in case of restorer gene, we have introduced this bar star system. This bar star gene, Barnes gene into male cell system. This is the basic. Okay. And what we have done, I will just show it here. See, this is the cross. Okay. This is Barnes bar cross. Okay. So what is this Barnes bar? See, for example, this bar gene is here, now Barnes. And this is bar. Okay. This is for that male sterility gene. Okay. But this bar gene is for gene responsible for herbicide resistance. Okay, here you see herbicide resistance. Okay, and this we cross with first. This is like the maintainer line. This is normal line, means normal, not normal plant, no transgenic, dash dash. Okay, but this is herbicide sensitive. Okay, when we cross, what will happen? It will be like this. Okay, on one hand it will have gene, other hand no. So the combination will be this and this, or be this and this, it will come like that combination in checkerboard. Okay, so what will happen? We will have genes like this: barnase, bar, dash, and dash and dash. Okay, this will be the product. Okay, so what will happen here? This is having this bar gene. Okay, so because of this, this plant will become herbicide resistant, and this is non-herbicide resistant. Okay, then what we will do? We will spray this phosphorythrin spray. That is, this is the herbicide which is sprayed on these two plants, plant one and plant two. This. Okay, what will happen? This plant, which is not having this resistant gene, will be eliminated. Only this will be there. This why we are making. You may ask why we are making all these things. Because because we need to produce this type of plants. Okay, we need to maintenance of male steroid means we need to maintain the male steroid. So this we are doing. No, nothing else. Okay, that's why we're doing. Then what we will do? This Barnes bar gene is crossed with bar star bar star. Means bar star is restorer. Okay. So once this is crossed, again, then he will have Barnes bar star, which is male fertile, and bar star, which is male fertile. So it will be 100% male fertile, and this system is very effective for increasing the heterosis. Okay. See, this is like a PYQ class. So this whole is like a whole concept which I've explained in 5 minutes. So even though maybe you have some doubts, you please go back and read or ask me. Understand this. Okay. So it will increase the heterosis, it will increase the yield. That's why we are bringing about this variety called TMH11 using this technique. If you write this technique, you will get mark. Otherwise, you can write these things, basic things also. But this is the what do you call the real content. Okay. So next, the question was about Mendel's law. Okay. Better define all the laws. Okay. Like the two alleles of a gene present in FN do not contaminate each other. Okay. The alleles present in F1 do not contaminate each other. They undergo separation and pass into different gametes. In their original form, okay, producing two different types of gametes in their original form. This is the concept of law of segregation. Next, we also need to know about law of independent association. What happens? Alleles of different genes. Okay, alleles of different genes will be assert independently, or the transmission of alleles of gene into a gamete do not affect the likelihood of the transmission of alleles of another gene into the same gamete. Like if you find it difficult, whatever definition you have read, that you can follow. Okay. Here you can see for the law of segregation, what should be the cross like. Okay, these things I'm little going fast because these things is very basic, which you would be knowing. Okay, so here you can see it will come like 3:1 ratio, dominant and recessive. Okay, and when you consider for the law of independent assortment, then we have this one. We have taking two characters into consideration. So here you can understand this law of independent alleles of different genes. Okay, the alleles of different genes assort independently. Okay, that's what in this, but here it is the same gene. Okay, so here you can see how these ratios and all come. These all things will be managed to 3:3 case of F2 generation. Okay. So next, there was also a question on cytoplasmic event. Okay. So what you have to write that all concepts you know that it is present on mitochondria plus chloroplast. and it is having uniparental inheritance. Okay, that is from the only mother plant, the inheritance will occur. Okay, these are also called like plasma genes based inheritance. Okay, so here also you can give some examples also. For example, in case of plasmid inheritance, case of plasmid inheritance, you can also go for different examples like the leaf variegation. Okay. And also in case of the mitochondria, you need to give an example. Like you can give the example of kappa particle in paramecium. Okay, these basic examples you can give, even a CMS system, all those things you can give. More than that, the major is characteristics. What are the characteristics? Okay, these plasma genes are associated with the mitochondrial or the chloroplast. Okay, that is the first point. Next, like they are having the uniparental, and the most important is reciprocal differences are observed. Okay, because what do you mean by this reciprocal differences? So if you're crossing A into B, or if you're crossing B into A, in case of nuclear genes, okay, there will not be much difference because uh, uh, even that regarding nuclear genes, whatever is father is mother is, even if you change father to mother, mother to father, that will not make any difference. But in case of cytoplasmic inheritance, if you change the mother, then the whole what do you say, the constitution of that the next generation will completely change. So that is the basic meaning, and there is no segregation in F2 and further generation. Okay, this is also very important to see. And also we find somatic segregation. These plasma genes show somatic segregation during mitosis. Okay, so these are the basic things which you should compulsory learn. Because I see students writing many other things, but they don't focus on important things. Okay, which we should observe. And there was also a question on chromosomal aberration. Okay, so this I have made, you can look at the YouTube video. I have completely described, it's freely available on YouTube. We'll just tell you basic only because you can see it very clearly. You know, due to mutation or spontaneous initiation, this structure may be altered. Okay, first you should define like what is chromosomal aberration, which is both combination of structural and numerical aberration. That is the rearrangement of the genes also may be changed, or the loss or gaining chromosome. Okay, rearrangement plus loss or gain. Okay, I'm just writing mention if you understand. Then when you structure and numerical, you can do it like this, or any sort of things when you get opportunity, like four common types. Make some diagram. Use your GS knowledge. Like in GS and all, we used to make diagrams, flow charts. Try to do that. I will also tell you. See, all these things I can tell you some other things, but it is you who will make it a possibility by thinking in the exam. Before that, you should practice it in the more for you. Okay. So in this, you know about how deletion, part of the chromosome of duplication is, part of the chromosome is duplicated, like DEF, DEF, you should make it like this. And in case of inversion, what will happen? The whole that chromosome segment is inverted. Inverted means ulta ho jata. Okay. What happens in case of translocation? It happens in what do you call it? Non-homologous chromosome. Okay, this is the keyword. I think you know. Homologous chromosome means which are pairing together. Okay, जो pair करता है that is homologous. नॉन होमोलॉगस मीन्स कुछ और क्रोमोसोम से इसका चेंज होगा. लाइक फॉर एग्जांपल ABCDFGH है, UVWXYZ है. Here if it is interchanged, then it is called translocation. Then we have concept of polyploidy. Okay, this also is there in the YouTube channel. Please go through this. Go through it. That would be much more be sufficient. Okay. So here, presence of more than two genomes in an individual. Okay. So you should try to. I told many students like to make it like this: auto polyploidy, allopolyploidy, auto allopolyploidy. Okay. Auto polyploidy, allopolyploidy, segmental polyploidy, auto allopolyploidy. Okay. Here the keyword is distinct genome should be there. Okay. This is the basis of like the polyploidy, and they've also asked about the application of polyploidy. So in case of polyploidy, so the major issue is about application because what students do is they write many application haphazardly, all those things. The major application issue that is one is like they create. There is a possibility of creation of a new species. Okay, for example, this Rapano brassica. Okay, I'm just writing all these things in short. Should note down. Okay, Rapano brassica. Okay, and also the other major application is the evolutionary relationship of crop is can be understood. Okay, evolutionary relationship of crop can be understood. The other major thing, like for example, if I give an example, like in case of wheat, okay, whether it is tetraploid wheat or hexaploid, how they work, all these things can be understood in it. Okay, then another is interspecific gene transfer. Okay, that is between species, the transfer of genes. Okay, so those things are also the major application of in crop improvement. And also the other major is like they are used as bridge species. Okay, while crossing different species, we use it as a bridge species. So you would know about Nicotian. Okay, in case of evolution of tobacco. Okay, we use the Nicotiana species. Like for example, first we cross this Nicotiana sylvestris with Nicotiana undulata. Also, we have then Nicotiana paniculatum and Nicotiana undulata. So such we can also use it as a bridge species. So these are the major things. Here also you can tell about triticale. Okay. So if you differentiate and write, then it looks pleasing to the examiner. The case concepts are clear and okay. And also you can write examples of that brassica. All those things you can write in this. These are the major part. Okay. So first, I'll ask the question about self-incompatibility. So first, you should write the definition. That is, self-incompatibility refers to the inability of plant with functional pollen which sets seeds when self-pollinating. Okay, so that is the basic thing about self-incompatibility. Okay, then we can also differentiate the types. Like for example, we have the homomorphic system and the heteromorphic system. Okay, so in case of homomorphic system, the incompatibility is due to what is the basis? That's what we need to write. Like the self-limited group which have the similar flower structure. Okay, similar flower structure, and it is governed by S alleles. But in case of heteromorphic self-incompatibility, okay, there are different types. It may be due to change in the height of the anther, okay, or it can be change in the pollen grain size, okay, stylar length, all these things are possible. Stigma. Okay, here we usually give the example that is distyly, tristyly. Okay, so you can give examples also, like in case of distyly, we have Linaceae family. In tristyly, we have families like Oxalidaceae, something like that. Any examples you can give. Okay, so that is like we have that pin and thumb example. So all those things you can give. After which we need to discuss about the gametophytic self-incompatibility and sporophytic self-incompatibility, which is very, very important. Okay, in terms of self-incompatibility. So what happens here in both, this is S allele, which is like having multiple alleles, is governing the principle in both. Okay, and this S allele is dominant allele. What happens in this gametophytic self-incompatibility? You should remember this. Please write down. Genotype of genotype of genotype of the pollen and pistil determine the incompatibility reaction. Okay, genotype of the pollen and pistil. Okay, will determine the self-incompatibility reaction. Okay, but in case of sporophytic, the diploid genome of somatic tissues of pollen product producing plant will determine the self-incompatibility. Okay, you are able to understand. Here, the genotype of pollen and pistil will determine the self-incompatibility. But here, the diploid genome of somatic tissue of pollen producing plant, the whole plant, okay, here the genotype, the plant is determining the self-incompatibility reaction. So you'll ask me how it is. The diploid genome of pollen producing plant is determining the self-incompatibility. Okay. So here you see, here there is, what do you say? The relationship is that the alleles are governed like this. Okay. Means this is more dominant over this and this. There are multiple alleles and it is governing by this principle. But here that principle is not. See, if it is same, it will not come. For example, S1, 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 S2. It will not lead to the pollination. But S3 can combine with S1, S2. Okay, because S1's effect is different, S3 effect is different. Okay, S1, S3 will combine here. Okay. In S3, S4, this is S1, S2. So S3 will also combine, S4 will also combine. Okay. So here you see there is a relationship that S1 is more dominant than S2, S2 more than S3. So what will happen? This relationship is not here. Okay. So what will happen? Even if it is S1, S2, it will work like S1. Okay. Here S1, S1. S1, S3 will work as S1. ठीक है, S3 का कोई काम नहीं है क्योंकि रिलेशनशिप ऐसा है स्पोरोफिटिक में. यहाँ क्या होता है आप बताओ. S3, S4 में क्या वर्क करेगा? S3 के it will be S3. The pollen will act like S3. Okay. तो S1, S2 क्या act करेगा? S1, S1, S2, S1, 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 S1. तो S1, S1 ब्लॉक करता है इसको. ठीक है. सेम जिन ये है तो इनकम्पेटेबिलिटी रिएक्शन है, यहाँ भी ब्लॉक है। लेकिन यहाँ देखो S3 है, यहाँ S1, तो उसका क्रॉस पॉजिटिव है। ठीक है, इसको दोबारा देखो, see this video again. Okay, so this is the crux. What is here? Here, the genotype of the pollen and pistil will determine the self-incompatibility. Here, they have individual effect. S1 and S2 have individual effect. But here, this relationship is there. S1, S2, S3 relationship. So this S1, S2 will act like S1. Here also, this S1, S2 will act like S1. So when there is this, like if S1 and S2 come together, then the cross will not happen. Okay, so cross will happen when S1 combines with S2 or S2 combines with S3. So it should be different. Okay, but S2, S2, yeah, S1, S1, that will not happen. Cross will not happen. That is the crux. Okay, this is what I can explain in very short manner. Should write all these basic points. Many of them they have written this portion, but this completely, let me tell you, most of you have ignored this portion, which is very important. Okay, if you don't understand, please ask me or first go through the book because these concepts are uh, like very deep. Like people may feel it deep, but this is the major thing. Okay, I think this topics also there are components of DNA. Okay, this is polyploidy. Okay, so this we will take up in the next session. Till then, all the best. So this will be class 3, which we will take it. Okay, okay, bye.